חונה גם השולחן שהיה פה. להעביר שולחן קטן משם הנה. כל עוד הם ילקחו לשם אחד מהשולחנות הקטנים בחדר השני. נשלח לכם את הלינק. לא יודע למי זה לכם, אבל...
אני פשוט רוצה לשמוע...
לא, אבל לאיזה אתר אנחנו נתנים? יש אתר לקורס?
כן,
יש מוד?
כן, אבל לא יודעת אם יש לך גישה אליו. אתה יכול לשלוח לי למייל.
עדי מזרחי.
אבל אנחנו לא צריכים עכשיו לשיעור.
תראי לי איך לעשות את זה.
אולי פשוט תשלח לי במייל, זה פשוט.
אני לא חושב שאת יכולה להכניס במייל כזה... זה יהיה כזה. רגע, אבל תני לי לחשוב שנייה אחת.
אולי דרופבוקס או... או שאני ננסה.
קודם כל נראה כמה הוא כזה. אני אראה כמה זה כבד שנייה אחת.
הוא לא צריך הרשאות בשביל להיכנס לניהול אתר באימייל?
זה יכול להתחיל 5 דקות.
אני לא יודע אם זה... אני אסתכל כמה דקות שנייה. $36 מגה, זה מה שאני לא יכול להביא. זה פשוט התמונות כבדות, זה הכל. אז רגע, אז מה אנחנו אומרים?
יש לך הרשאה, עדיף ידע לכולם.
איך אני בודק אם יש לי הרשאה? מה אני צריך לעשות?
אם תיכנס למודל לאתר הזה...
מודל. את רוצה לכתוב על זה תוך כדי זה, זאת אומרת? למה את עושה את זה עכשיו בעצם?
אני פשוט אוהבת לסכם על המצגת תוך כדי שלה.
בנוטס למטה או מה?
אצלנו יש משהו אחר.
אני אגיד לך מה אני עושה, נתתי הרצאה דומה, יש אולי כמה שקופיות שונות, שקפים שונים חדשים, אבל ההרצאה דומה נתתי בסמסטר ב' של השנה הקודמת, בקבוצה שלפניכם. וזה יש לי לינק. ושוב, אני לא בטוח שצריך להוריד את זה מהרשת. את תוכלי לנסות, אבל זה ייקח לך... אוקיי, בסדר. אז רגע, גם צריך להיכנס, בשביל לראות את זה. להיכנס ל... פאבליק לינק, לאן לשלוח את זה? זה אמור להגיע אליי, אני מקווה. אוקיי, עכשיו נתחיל הגיע אליי? אולי יש כאן בעיית אינטרנט, אני לא יודע, זה נראה לי שהוא שלח, הוא הראה לי שהוא שלח, אבל לא. זה לא הרבה לכתוב במיוחד. אוקיי, טוב. שלום. כן.
Can you bring the microphone?
Sorry?
The microphone? It's on.
Really? Can't you hear me?
It's also for the video if you want to watch it later.
Okay. Should I speak English?
Yes.
That's the main thing. Okay. Okay, fine. So welcome to the ICNC, so to speak. So we have 4 meetings. 3 will be discussed by me. Mostly I would like to represent, mostly intuitively with some equations, but mostly intuitively, how I see what we can call a single neuron computation. So you know that neurons are rather complicated. We'll talk about them. You know about them a little bit. I'll try to give you my long-term intuitions about these unique characters, these neurons. And then I would like, after talking a little bit about how I see neurons, and I like always to give a little bit of a perspective about neurons, so that you don't think that we started yesterday to discuss neurons, although it's not long time ago, absolutely not long time ago, that the term neuron came about. So it was not clear at all that there are neurons in the brain, about 120 years ago. So I would like to start with a little bit of this Cajal-Golgi controversy, just to give a perspective about neurons. Then I want to give you some very recent angles about neurons, some new facets about neurons. But again, one thing I should ask you to do is, first of all, interact with me. Don't sit and just write, but listen and also interact. But also tell me that you know it. So, because there are so many things to do, and I have so little time. So if something sounds, like familiar, so let's tell me, this we know, we heard, we would like you to move forward. So I will do that. So it's a little bit of an interactive kind of 4 hours. In the last hour, in the last session, the last one, the 4th one, you will present, 3 of you will represent 3 papers related to this topic, a very recent set of 3 papers related to neurons as computational units. So that's the plan. I will start with passive cable theory. Again, you heard a little bit about passive cable, so I'm not going to develop the cable theory, but only to give you insights. I see myself as insightor for you, because after many years of working on these problems, I can give you insights that are very hard to acquire. And insights, of course, are the most important things after you integrate data, some insights. So I'll try to provide you my insights about certain things along the way. But maybe before that, just to give you a more perspective, I don't know if you are aware of it, but you are sitting really in unique times in terms of brain research. You should know about this. I guess you heard about Barack Obama recent announcement of his neuron brain mapping project. Something I should also put in the doodle, really. I have to remember it, because I have now, it's not yet published in an official way, so you don't really know exactly what Obama wanted, although we shall talk to him in 10 days here, and we'll see. But maybe he doesn't know himself exactly what was this announcement. So, but it's an announcement about basically that we would like to be able to develop methods to record, and the question is, what does it mean to record? But to record the activity of all the neurons in the brain, which is very dramatic, of course, because you know that today we can record or look at the activity of 1,000 neurons simultaneously, but we cannot record from a whole region at the level of single neuron. So that's the American ambition. That's the landing of the man in the moon, on the brain from America. And the European claim, which I'm part of it, is what is called the Human Brain Project, was announced on the 28th of January. This is a €1 billion project. I hope maybe in the third lecture, if time will permit, I will tell you more about this Human Brain Project of the European brain. But it's a very special time, really special, and not coincidentally, these two continents are fighting about the brain, because I think it's a reflection of the fact that we don't understand the brain. There is no V on anything, which is terrible. There are more and more diseases, and the pharmaceutical companies are leaving the brain. They are leaving brain research. These four things, and of course there are new technologies that are dramatic and fantastic. We should mention a few. So this kind of combination of things, the fear of the Homo sapiens, that the brain is getting older, that you cannot understand it until today in a real deep sense, I think is one of the fundamental reasons for this dramatic new project. But we can discuss it later on. So this is just a real introduction to, for me especially, a very, very special time, because we were hoping for these moments to come. Whether they will be successful, whether we shall do something new because of these dramatic claims, I'm sure we shall do many new things, but I mean, whether it will be good enough, it's for us to prove. Nobody else can do it. I mean, Obama can give the money, but somebody has to work, you know. Okay, so perspective. But again, ask me questions and don't allow me to talk an hour and a half here, which is possible, but it's not recommendable. So it all started here, you know, with this Nobel Prize, this year of Nobel Prize to these two characters, which you should be friends with, because without them, and without the big controversy between the two of them, and I didn't bring here, but if I had more time, I would give you a whole lecture about this very interesting controversy between two giants. There are giants, anatomical giants. I mean, two real giants. I mean, the best anatomy is anatomy ever in terms of what they did to the brain, but they did agree on one particular thing, you know. Golgi, the Italian fellow, the romantic guy, not that he was not romantic, but he was a very romantic. He did not believe that brain as other tissues are built from cells. He could not think that our, you know, emotions and feelings and creativity and, I don't know, whatever you do is constructed from units. Unlike what they already knew, that, you know, the skin is built from cells, and the muscles is built from cells. But the heart is built from cells, but not the brain. That's what Golgi thought. So he thought it's a continuum, it's a continuous, he called it syncytium, he called it some continuity of a network, not of individuals, and this network subserve opera. He was Italian. And Ramon y Cajal looked at the nervous system very carefully with his very primitive microscope, using Golgi's staining method. So Golgi developed a staining method for the brain, so he could see individual structures, but he didn't call them cells because he thought they touch each other continuously, so to speak. But he developed this Golgi technique, and Cajal was using Golgi technique to look at his preparation with some improvement that he himself did, so enhancing Golgi technique. And he saw something, and he was sure that not being able to really see the split, the synapses, there was no term synapse at that time, but he saw that there are individual cells. So this was the big controversy. And on the Nobel stage, on the Nobel podium, he was holding for his view, saying,
it's a continuum. My colleague, my friend, my respectable friend is wrong. And he was saying, my colleague, my respectable friend, I'm using his techniques, but he is wrong. So today we know who was wrong, or who was right. We know that he was right. So he was right in terms, and that's a very important point to point, he was right in terms that in the central nervous system, not always, not everywhere, but in the central nervous system, there are this, what is called synapses today, a gap, discontinuity, physical discontinuity between neurons, and so that you can, so to speak, kill one without killing the network, if you want to call it this way. They are disconnected. Somehow they transfer something between them. He realized that it has to be community work, but of individuals. So somehow they should talk to each other, like we do now, but we are still individuals in a physical sense. It's not always true. You know that there are electrical gap junction or electrical synapses between some cells, also in our brain, in the retina. And of course, when you go to invertebrates, you see a lot of electrical synapses, so it's not always chemical synapses. But he was right in that sense. And it was very important in many ways, because you can ask who cares if it's continuum or not continuum. It's a nervous system. Of course, it is very important both for, first of all, knowing what's happening there, but also for many, many theoretical issues, and also pharmaceutical issues, because, you know, if there are synapses, maybe there are chemical aspects, maybe you can control the connection, maybe you can learn through connections. Of course, the fact that you have synapses and cells and interaction between them has a lot of functional meaning. It's not just talking whether they are connected or not in an intellectual sense. So Cajal was a big winner. Personally, I think he was in effect, you know that Van Gogh at that time on south of France, in Arles, before he cut his ear, Van Gogh, was painting beautiful trees. And so painting trees was in this era, in this area of time, but this is my personal association. I don't know if it's true or not. He was painting, the trees became something very beautiful, and Cajal was an absolutely fantastic artist, as you know. So these are slices that exist today in Cajal Institute, in Cajal Museum, in Cajal Institute. Today you can see Cajal's original cells. When I say Cajal original cells, I mean staining of Cajal using Golgi stain, and then reconstructing it by eye, looking at it and painting, you know, drawing, process by process by process by process. He could not see it in such a very good resolution. This is new microscopes, but he did some amazing things, and this is some of his amazing drawings, which you know. Yes?
This Golgi you mentioned, is he the one that discovered the Golgi apart?
Mm-hmm.
With the same instrument?
No, different technique. I don't remember the timing. I don't remember.
But I just wondered how
I don't
it's
remember.
possible to discover something that small which is inside the cell,
Mm-hmm.
if, as you said, he didn't
The resolution. I have to check. It's a good point. I have to check it. I never thought about this resolution issue and also the timing. Let's wait for it and look at it in the literature. I'm not sure. So hold my answer. We'll discuss. I think so.
Were most of Cajal's works in human cells, or were they animal cells?
So this is a good point to discuss Cajal. Cajal was, as I said, a genius in many ways. Very controversial, unusual personality, really, really very unusual. And he actually tried to encompass, and I don't know if there are these two huge volumes of original Cajal that he wrote, and he analyzed what he sees in every animal that you can think of, so to speak, yes? Elephants, giraffes, humans, rodents, whatever he could reconstruct, he reconstructed. He liked to paint. He wanted to be actually a painter, and eventually didn't work out. So he really did amazing work, really, really amazing work. Not only that he has a very nice, capability to detect through the microscope. You understand that he looks at this microscope and he does this, but in such a fantastic way, a fine way. But also, while seeing things, he saw some things beyond. For example, he saw that there are individual cells. You cannot see it. cannot see individual cells. You see a network. How do you know that this cell is yes or not connected physically or not? So he invented some things in some sense. He thought that he sees something. He had some arguments because he could kill one cell sometimes and he see that it's not the whole network, it dissolves, so maybe he had some indications, but he could not prove. Only when we had EM microscope, which is in the middle of the 20th century, we could really see the synapse and we could really see the gap. Until then, but he was a very creative brain, very creative, also very good artist, very prolific. In some years, he published 14 papers on his own, and because nobody accepted 40 papers, he published his own journal suddenly, the Cajal Journal, with his own. He was not a regular guy. Of course, we need people like him. So these are beautiful drawings, and they are classics in many ways. You know, these Purkinje cells are really absolutely fantastic. You can see them almost in every, I don't know where. And so you see the retina. He reconstructed the retina, and he saw the receptors, and he saw the bipolar cells, and you know, the optic nerve. This is all 120 years old, and also with colors. So it's beautiful. So there is a book, if you're interested a little bit with the history, and I don't want to continue with it. Just to tell you that there is a book that summarizes these things, the history of the development of ideas of the neuron doctrine. It's called The Neuron Doctrine. It's a book by Gordon Shefford, who is an anatomist himself. And he really speaks about the development, including Freud, not Lucian Freud, the painter, but Sigmund Freud, who was also an anatomist or physiologist at a certain time in Trieste, Italy, and also drew cells. And he also thought that cells are individuals. So Freud was on the side of Cajal, and there were others on the side of Golgi. So this book, The Neuron Doctrine, if you want to look at it, you should look at it, because there are also beautiful drawings there, but also the history of ideas. One point about Cajal, and we shall discuss it later, because here he was mistaken in some sense, is that he drew these arrows. This is the original arrows by Cajal. And he drew these arrows trying to show, and he called it the theory of dynamic polarization. He said that the cell is polarized. Again, this is all from anatomy. There is no recordings. There is no electricity. It's all from anatomy. He says, okay, there is something that I called an axon, from the thalamus in this case, thalamic axon, climbing, so to speak, into the cortex. And there is something that is jumping, I don't know what, jumping, connecting, although because they are not connected, yes, as we said, jumping, this is the arrow jumping information, electricity, I don't know what, something is jumping from one side to the other. So the dendrite is polarized. First here, and there is a flow of information, as this arrow indicates here, and then there is a flow of information from this axon to the dendrite of the other one. So the concept that you are all built on, we all are built on, is from axon to dendrite, from dendrite to soma to axon to dendrite. Yes, so this direction of information flow is Cajal. This is what is called the theory of dynamic polarization. Okay, so the neuron doctrine is his. The term neuron was established later by a German guy, not by Cajal. And the polarization, that somehow the information flows this direction is also this concept of Cajal, but based on anatomy. You could say that it goes this direction. Why did it go this direction? But he said, it comes from the eye sends axons. There is the optical axon. It must go through this. So there is a direction of flow of information, which there is in some sense. But that's what Cajal said. Okay. But today, of course, we can record from dendrites using these new technologies. And this is, optical imaging. We shall talk a little bit more about that. We can record from dendrites actually today quite stably, which is relatively new for neuroscience, to be able to record electrically, not optically, electrically, directly with an electrode in dendrite, try to see this unique structure of neurons, which is so beautifully you saw before, this unique structure, what kind of electrical signals are happening there. And this is something we could not do until, systematically, we could not do until, let's say, 5, 7 years ago, 10 years ago. And now, we could record from the cell body, which is relatively big, 20 microns in diameter, and then most of the information that you still see today, and most of the modeling that you still see today is from the somatic point of view, somatocentric, as if we sit in the soma. Why soma? Because there is an axon, and the output is in the axon, so soma is important, of course, but interesting things are happening, computational things are happening in the dendrite. Not only most of the inputs are, of course, synaptic inputs are into the dendrite, but interesting signals interesting nonlinearities are happening here, and that's what we want to discuss. What kind of electrical nonlinearities are happening in the dendrite, pre-processing before there is this output coming from the axon. So we have new tools in recent years. This is only one of them, of course, to look at dendrites in fine resolution and record the electrical activity, both optically but also electrically, as you see here. We can also reconstruct neurons, and if you had special glasses, you could see this axon coming to you. So these are reconstructed, fully reconstructed, and I didn't want you to see things coming through, but you can go to, I don't know, to the internet and see. But what I did want you to implant, if you didn't yet, is that although the dendritic tree, which is this green thing, this is a pyramidal cell in the cortex. It's by Kevin Martin from Zurich. So the green thing is the dendritic tree. This red thing, the red tree with its puncta, as it is called, or bouton along the axon. So this is the axon. These are axonal branches. You can see the bushiness of axons. So in your cortex, of course, most of the wiring are axonal wiring in terms of length. Dendrites are rather local, maybe millimeter long, something of that order. And then the axons can be really very, 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 very intensely bushed, so to speak, around you, but also often, of course, can send, and this is cut, can send, of course, interaction from one hemisphere to another, or from one hemisphere to the spinal cord. So this could be very long. And you can see these little yellow points, these boutons, kaftorim, along the axon. These are the release sites. These are the release sites, presynaptic sites, where you have all the machinery with the vesicles and everything that is needed to be released upon the action potential propagation along this structure. And there are maybe 10,000 release sites per axon in the cortex. So you talk to 10,000 friends from this release site. So just to give you the complexity. If you want more numbers, for example, if you look at the cortical column, let's say that this is your skull, and this is 2 millimeter deep, and this is 1 millimeter wide, and there is 1 millimeter deep, so it's about 2 cubic millimeter. On average, in the cortical column like this, you will get about, depending what species, let's say in the mouse, you will get maybe about 20,000 or 30,000 cells within the 2 cubic millimeter cortical column, as it is called. So about 30,000 cells altogether. And we'll talk about the Human Brain Project. You'll see how we are trying to make sense of these cells, type of cells, inhibitory, excitatory, layers, and all that. But also, just to give you dimensions, so there is about 4 kilometers wires within 2 cubic millimeter. 4 kilometers of wires. Can you think? About 4 kilometers wires within a cubic millimeter. And there are maybe in our brain million columns like this. So you can see how many times we can, with our wires, wrap the world, wrap the Kadur Haaretz, yes, how many times. So anyway, and most of these wires are axons. And within these axons, about every 4 microns, you see a release site, a release site, a release site, talking to somebody else here that you don't see, another cell. So this should give you a little bit about the jungle in your brain. Okay, so we'll talk later on about this relatively recent finding about propagation of action potential backward. I'm not going to, did you hear about that? You spoke a little bit about that? This came from this type of work where you could record simultaneously from different locations in the same dendritic tree of a given neuron. You could see signals propagating forward, synaptic inputs, local calcium spikes, but also back-propagating from the axon soma region back to the dendrites, which is a revelation. This is anti-Cajal, who thought always on this one-directional flow of information. This is reverse, so to speak, what is called antidromic. This is called autodromic, antidromic. But we'll talk a little bit about that one. If you heard a lot about the action potential, then we won't talk a lot about this. And one thing we are not going to talk a lot about this course, but last year I gave it as a special paper to read, because I thought, is it working, by the way? Yeah. Because I thought this is an absolutely amazing jump in terms of technology. So did you hear, all of you, about the work of Connors? Okay, so this, to me, if you ask me what happened in the last 3 years, let's say, in neuroscience, that in my mind is a jump, not only technologically, but really opens a new set of questions very relevant to the questions that I'm interested in, single neuron computation, or some of the questions I'm interested also in network, but for this course, for the single neuron, is this amazing capability that was developed in Munich by Arthur Connors, who comes here very often, very, very technological jump, and that's the following jump at the single cell level. So Arthur Connors works with mice, and he records from the visual cortex of the mouse, V1, primary visual cortex of the mouse. He can show this mouse movies, moving gradings, this direction, this direction, this direction, the classical Hubel and Wiesel, let's call it. But in this case, he can record not only from a single cell, which is classical by now. So this is now the cell. This is in vivo. So this is anesthetized, but in vivo neuron, this case layer 2-3, in V1, a layer 2-3 pyramidal cell. He can record with an electrode, classically as it is now being done for many years, in vivo. So you blindly, so to speak, seek for, or not blindly, today you can stain the cell and you can find this cell body. You can impale with an electrode here, but, and this is a jump, I mean technology, at the same time you can image at this point, at this point, at this point, at this point, and many points, in this case not so many, but today with more points, you can image the activity or the receptive field, I should say, of the input to the cell. So you record, for example, let's look at this synapse, I don't know, this synapse, let's look at this synapse. So optically, using two-photon microscope, which I hope you heard about a lot. Did you? Yes or no? It's fine. You can tell me no. I don't. You should tell me what you know, and then I know what to tell you. So two-photon microscope is Adi Mizrahi is our person with two-photon microscope. Has this capability to zoom in, still at the light microscope resolution, using laser technology. And Adi, I'm sure, will tell you when he tells you about. Or I should tell him to tell you if you don't know about this technology. It enables us. Elin Elkin is working with it. Mickey London at the ICNC are working with it. It enables us, until recently, before this paper, to detect calcium signals which are related to the activity of the cell. So when the cell fires a spike, there is some calcium flow into the cell when there is an action potential. And if you have an indicator for calcium, which absorbs, so to speak, calcium and project fluorescent dye following the entry of calcium into the cell, then you see the cells is lighted up, so to speak. Whenever there is an action potential, you see some fluorescent coming from this cell. And so this enables you to optically zoom in to the level of the cell body of this particular cell. And instead of recording electrically, you record optically the activity through calcium flow, which indicates activity of spike in this cell. So this is now classics. What I told you is about now 10 years. But this jump, which is an improvement, which I don't understand exactly the technology, enables you to zoom in with two-photon microscope, not only to the cell body and see spikes, but also see synaptic activity at the postsynaptic region of the dendrite. So what they actually do in principle, is to increase the resolution of the two-photon microscope. So if there is an axon going here, making, let's say, a synapse there, and if I zoom out, I can see that this is the dendrite, and there is a spine here. This is the postsynaptic spine. The axon is going here, somehow making a synaptic contact, so the vesicles. Okay, and so when this axon is active, this spine develops an EPSP, which you cannot record. We cannot record electrically from individual spines yet. But we can now record optically using two-photon zoom into the level of single spine. You understand that the single spine is, let's say, 1 micrometer square in area. The soma is much, much, much larger, let's say 2,000 micrometer square, so it's much easier to record activity, also the calcium activity in the soma, than in this little soma, which is not a soma, it's spine head, but that's what they did. So they succeeded to increase the resolution of recording to the level of the input to the dendrite. And they asked themselves, what is the receptive field of the input to this dendrite? Do you understand what I'm saying? The receptive field, what is the directionality? So for example, when he says, let's look at this synapse. When I said this synapse, I mean the activity of the postsynaptic spine due to the activity of the presynaptic synapse. That's what I mean, the synapse. So when I'm looking at this synapse here, and I see that there is calcium flow into the spine only for this angle in the world. And if you look at this spine, I don't know, this spine, you see that this spine likes these angles in the world. Okay, when I look at this spine, so these are the receptive fields of the input to these spines onto the mapping of this dendrite. So this means that this postsynaptic dendrite, this layer 2-3 dendrite, is seeing all angles, if you want to see it like this. So it receives input from this angle in the world. And this synapse is representing this angle, and this synapse, this angle, and this synapse, this angle. But eventually the cell is responsive to one angle. So you understand that there are axons coming here, and these axons are from other cells in the network. It could be layer 2-3, it could be from layer 4. We don't know we don't know who is the origin, who is the father of this synapse, or the mother of this synapse. But we know that the input to this synapse, the axon talking to this region, likes the presynaptic cell, likes this angle. This other presynaptic cell likes this angle, yes? This is all excitation, yes. So that's a limitation of the technique, because when I speak about spines, we'll talk about spines and about inputs to spine, and especially about calcium flow, it's always from excitatory synapse. Yes, you increase, you depolarize the membrane, you open calcium channels postsynaptically, there is flow of calcium, so you cannot detect, and this is a big myth, we don't know what is the orientation selectivity or the mapping of inhibitory synapses onto this same cell. So we don't have the complete picture of who is talking to you in terms of angles. But one interesting thing in this case, many interesting things, but one interesting thing is that this particular cell, which is eventually a directional selective with some breadth. Yes? So let's say that this cell has a directional selectivity of 45, I don't know, angle of 45 degrees, plus minus something. So there is a receptive, this you know, when I say receptive field, is that clear? Or so there is some tuning to this cell postsynaptically. Let's say that this cell postsynaptically, the tuning, so this will be now angles. So this will be an angle in the world. And let's say that this cell has a tuning curve like this. I don't know. Let's say this is 45 degrees with some width. So this particular cell is one among these other cells which has a particular orientation selectivity, but the input to these cells is coming from all orientations. This we did not know. Okay, we could not know before who is talking to me. don't know who is talking to me. I know only when I record for the cell body that I'm saying 45 to the output, but I don't know who is the input. So in my mind, this is a huge jump, because one open question is, how do you compute, taking all these angles, deciding, so to speak, that I'm 45? Because you have also 30, you have also 37, you have 42. How do you decide that you are 45? You see everybody, so to speak. So we don't know what is how the computation is being done. It could be simply by majority. I don't know. Maybe from the 10,000 synapses, 8,000 are 45, so they are taking over. It could be some nonlinearities locally. I don't know. It's an open question.
Do you have any indication of the strength of each?
That's a good point. That's a missing point. Although there is some correlation between the size of the signal and the strength of the synapse, but not exactly one-to-one, because it depends also on the area of the spine, so how much membrane area. We have other indications of the strength by doing EM, looking at the postsynaptic density, the amount of receptors on the spine head, but that's another missing information. So it could be not by majority in terms of number, by majority in terms of strength. And this I cannot tell you directly from there. But it's a big, so you understand, okay, so I don't want really to dwell into this, because this comes more to the computational aspect, which is the end. I just wanted to highlight a technological jumps, because one of the main open question, of course, is how this network eventually compute, let's call it, or how this cell eventually compute a particular angle. What makes it responsive particularly to, let's say, 45 and not to 30? We don't know. There are models. You know, there is Hubel and Wiesel model. There are other models. But until we really go into the level of this level, and even more level, because you want strength, you're right. You want inhibition, you're right. I really don't understand how this microchip compute orientation selectivity. There are ideas, a lot of models, including that of Chaim, which is a very beautiful model, but we eventually have to go there. There is no way other than that. There are ideas, but this will eventually pin down this idea versus the other idea. I will show you on the third lecture that there are very recently, maybe you've heard, I don't know if you've been here, some of you may have, to the lecture of Mitya Chlovsky from Janelia Farm, who looked at the retina, reconstructed retina. I thought it was a fantastic talk. I knew this work and related work, so this is in the retina. Also there you have directional selective ganglion cells already at the retina. This is already the cortex, so there are so many stages. But still, how does the cortex compute orientation selective? I just want to highlight before I go on that this is a mouse cortex. Yes, it's not cat. In the cat cortex, this is Hubel and Wiesel. In the cat cortex, Hubel and Wiesel found, as you know, that what they call the orientation column, the whole column that I spoke about before, the whole column of this 2 cubic millimeter, the column, all of the cells there, or most of the cells there, respond to the same direction, orientation. Most of the cells have the same orientation selectivity. In the mouse, and this we knew already before because we could record from the cell body of this cell, of this cell, of this other cell, this other cell, we see what is called the salt and pepper organization. The salt and pepper, which means that nearby cells may have very different orientation selectivity, which is a different functional organization than in the cat. Still, the mouse sees, not as good as the cat does, but he sees, otherwise he cannot navigate. So you may be able to see with these organizational principles the salt and pepper organization rather than with a columnar organization. But still, it's a different organization. So in some sense, you would not be surprised. I was not really surprised by seeing the fact that this input comes from 45 and this other input comes from, because nearby cells, we knew, have different direction selectivity. So if they have an axon connected to this dendrite, it means that you will find the salt and pepper organization also on a single dendrite. So this salt and pepper organization that I show you now is a reflection, so to speak, of the salt and pepper organization of the network activity. But still, the question is, how do you eventually become 45? It's a question. Is that clear what I'm trying? Okay, so this is an interesting organization. Actually, I have a student with Chaim Sompolinsky, Yoav Tal, who is trying to understand something that is quite strange, that you have in a volume, very packed volume, as you saw, very, very packed volume, many, many cells together, sitting, talking to each other, axons and all that. And suddenly, two nearby cells say something very different. You would expect that there will be some kind of an averaging out. And I will become the same like you and the same like you because we sit in the same environment, we listen to the same axons. So how do I become an individual? Different than you that sits near me? Not clear. What do you need to assume about connectivity? That eventually, or about computational dendrites, or about some other mechanism that will give you this separation, this singularity of each cell saying something else. Remaining individuals, although we are in the same company. I don't know. Another interesting thing that is being said here, I think, and if you will ask Yossi Arom from the ICND, he will say, this is an example showing that you cannot tag a cell and say this is 45 degrees cell. Because now it's 45, maybe tomorrow it will be 30. Because he has all the inputs already, you just make this one stronger, this one weaker, maybe inhibition in here, I don't know what, and suddenly it becomes responsible for 30 degrees because it's there. It doesn't have to learn much. The connectivity is there. It's only a matter of who is winning. Maybe right. Maybe right. In this case, in the column of the cat is something else. The column is a column. You can force the column to change directionality after plasticity and learning. It's true also in the cat. If you show a cat all her life, its life 45, eventually all the columns will be 45, at least in the critical period of time. So it's not that it's hardwired to be 45, also in the cat, but after a certain point, probably in our case, this is 45 and this is 33. What can I do? I can look all my life at 45. I'm not sure that everything will become 45 now. But when I was a child, maybe. But anyway, this is a child, so to speak, mouse, because in the mouse it's always capability. The capability is always to switch. What does it mean computationally? Is it better or worse to see the world and so on? As I said, the mouse is not such a visual animal like the cat. Okay, so but this is a real, for me, jump, because for the first time I could also detect the input, not only the output, and try to correlate structure to function, or input to function, if you want to call it this way. Okay, what do you say about my following suggestion? Can I talk another half an hour and leave you, or do you want a vacation, a little bit of vacation to drink? What do you prefer? I don't care, actually. אתם רוצים הפסקה, a break? A break. But not long, okay? Because we started a little late. So let's take 10 minutes break. Is that okay?
זה הרעיון של הסיכום הזה, שאפשר להקשיב. אני לא יודעת אם אתה מתעסק רק לנקודות של האוניברסיטה, שאתה לא יודע.
מה אתה מצפה מ-08?
לא, כאילו, אתה לא חייב להקשיב, זה טוב.
אוקיי.
אני כזה עושה טגים.
לא, בכל מקרה זה טוב שיש את האופציה הזאת, אם לא מבינים משהו.
זה בסדר. אני אומר בעד.
10 דקות.
כן. כן, סליחה.
אני מציגה אחד המאמרים על אלסק שאני לא זוכרת שראית.
תזכיר לי, כי אני גם לא זוכר. כשאתה אומר לידר זה לא אומר לי.
נכון.
אני צריך לשם.
אז יכול להיות שיש לי. בכל מקרה, לא, האמת היא, פשוט הבנתי מידי שכדאי בכל מקרה לקרוא קצת מאמרי רקע בתחום. אז רציתי לדעת אם יש לך איזשהו review
אם תגידי לי מי זה ה-Letter to Nature, אז אני אגיד לך מי הרביואים האלה. או, את יודעת מה הכי טוב יהיה לי? תשלחי לי. תשלחי לי מייל. תגידי לי, זה המאמר שלי.
ולבקש איזשהו חומר רקע. תודה רבה.
המוח הם מאוד מאוד נהניתי מהם. ככה למדתי פסיכולוגיה ומוזיקה בכלל. לא, בחיפה, למדתי שם אוניברסיטת חיפה. מוזיקולוגיה ופסיכולוגיה? לא, למדתי מוזיקה. למדתי אופקים, זו תוכנית כללית של מדעי הרוח, של לפני זה למדתי בבית ספר למוזיקה. ככה היה לי מסלול קטנטן מאוד,
והם הצליחו לקבל אותך, אתה אומר, זה לא קל.
כן,
יכולת ללמד משהו ממש.
כן, פסיכולוגיה, זה לא חלק רלוונטי. זה היה מאוד מאוד מעניין. אני לא יודע אם זה טובה שאני חייב לך הוראה, אבל כן,
אני חושב טובה. לא חשוב מה יצא, חשוב שנהנית. אני חושב, לא?
כן,
אני יודע מה היה אחר כך.
זה נכון.
אני יודע מה היה קודם.
זה נכון.
אוקיי, טוב. אנחנו רוצים להמשיך בענייני תאי העצב, או העצב צריך להגיד בעברית, אבל אני לא יכול להגיד רהוט בעברית, כי אני לא יכול להגיד עצב, זה קצת קשה לי.
אתה מדבר אנגלית.
ואני מדבר אנגלית. You're right, I forgot, I have to speak English. I was joking a little bit about Hebrew idioms, that nerve cells should be עצב, but we all say עצב, and עצב means sadness, so it's a problem here. But I'm going to speak English, so we don't have problem. Okay.
You have a microphone?
Okay, so salt and pepper. We were on the salt and pepper issue. And this is just a summary of this paper, which, as I said last year, we went through very deeply. What is known today, at least in layer 2-3, both in the visual cortex, also auditory cortex, which is the work of Eli Nelkin with Arthur Connerth, and also in the somatos whisker system, is that there is this mapping, which is an intermingle mapping. That it's not a one dendrite having all the orientation and another dendrite having the other orientation. It's a mixed orientation. So within a given dendrite, you can step, so to speak, along the dendrite and find different orientation within a single dendrite. So it's a kind of a real mix. And as I said, we don't know how come that this mix of inputs in terms of orientation selective eventually generates particular output orientation selectivity. Okay, so because dendrites are so fundamentally important, because most, if not all the inputs, not all, most of the inputs are going onto the dendrites, at least in the central nervous system, we can speak about different properties of dendrites, which will be in the internet, which will be there. You don't need to now remember everything. I just want to highlight what I think is something that you should know. You cannot be a neurobiologist without knowing it. Maybe you already know it. So let's see. So in terms of morphology, in terms of basic dimensions that you should remember, I think, is that the total length of dendrites from soma to the tip of one dendrite, not if I take all of them, assemble them together, but the total length from cell body is on the order of a millimeter. Of course, there are short dendrites, there are a little bit longer dendrites. For example, layer 5 pyramidal cell has a very long apical tree, which could be maybe 2 millimeter long, but on average, 1 millimeter is a good size of a dendrite, okay, from cell body to the tip. So this is something you should remember, I think. The diameter of dendrites, of course, changes from the cell body, near cell body, which is thicker, then it becomes thinner as it branches out. But a good diameter to remember is on the order of 1, 2, 3 microns coming from the cell body, and it could become as thin as half a micron, 0.3 micrometer distally. And maybe I should also mention that the area of a dendritic tree in terms of total area, and your brain is mostly dendrites in terms of area, not in terms of length, because I told you about length, which are the mostly axons. But axons are very thin, and dendrites are thicker on average. So overall, you get more area on the dendrites, maybe 60% in the cortex, and 40% area are in the axon, but an area of a single neuron on the average. Again, there are small spiny stellate cell, big pyramidal cell, average pyramidal cell, so I'm saying something very crude. But on average, you should remember something on the order, let's say 20, 30,000 micrometers square. Some dendrites are huge. Some dendrites have small area. But let's talk in the middle, somewhere 20, 30,000 total membrane area. So this is the character you are talking about, yes? It has a 1 millimeter long, maybe many dendrites, but from cell body to the end of one. Sometimes you have 10 branches coming from the cell body. So in this case, you will have 10 millimeter long. So it depends on how many dendrites, but one is about one extension of one, so it's what we call local, because dendrites do not go over other locations and far away. Axons do sometimes. And some dendrites are spiny, and some dendrites are smooth. And mostly, not always, but in the cortex always, whenever you find a spiny dendrite with these little spines popping out, and many of them, this is an excitatory neuron. I don't know why. Why a spiny neuron in the cortex is also talking excitation to all its output neurons. But there is a correlation between the spininess of the neuron, of the dendrite, and the fact that this neuron is excitatory. It's not always the case. For example, Purkinje cells in the cerebellum, these beautiful cells that you saw before from Cajal, they are very, very spiny, densely spiny. 200,000 spines within a neuron, the largest number I know, but the output is inhibitory. So it's not always the case, but in the cortex, there is a correlation between the fact that the dendrite is spiny and the fact that the output is excitatory. And the smooth cells in the cortex without the spines are inhibitors. So, and when you have spines, there is a lot of area in the spines, because there are so many spines, maybe 10,000 spines within a cortical pyramidal cell, and each spine is 1 micrometer square in area, so you have 10,000 of them. So it's a significant area of the dendrites are in the spines. And that's very important for understanding plasticity and connectivity. The spines are important in terms of total area in pyramidal cells, for example, or in other cells. So this is morphology. We are talking also, we are characterizing neurons, of course, by their physiology. We speak mostly in the beginning on the skeleton, which is passive skeleton. We can talk about the passive skeleton, although neurons are never passive. But I can characterize them, and that's cable theory. Characterizing neurons using the passive properties of the neuron. For example, I can speak about the resistivity of the membrane per square centimeter. If I take a membrane, square centimeter is huge. But if I had a square centimeter of this membrane, of the dendritic membrane, I can characterize what is the resistivity of this piece of membrane. So I speak about Rm. I can speak about the capacitance, specific capacitance per centimeter square of this membrane. And so these are electrical properties, the passive or the skeleton or the basic electrical properties upon which you may add excitable channels, nonlinear channels, voltage-dependent or synaptic-dependent channels. So this is the passive skeleton of neurons. And just to give you a ballpark numbers, for example, Rm, this is this membrane resistivity for a square centimeter, is on the order, let's say, in cortical cells, in vitro, quiet neurons sitting on a dish, it will be around 20,000 ohm centimeter square. Note the units, ohm centimeter square. Capacitance farad per centimeter square. And this is ohm multiplied by centimeter square. And I don't want to go into it, but if you don't understand why sometimes it's multiplied and sometimes it's divided by centimeter square, we can talk about this. Then the important thing is that when you multiply Rm by Cm, by the way, this universal number of membrane capacitance for living cells, which is the property of, the dielectric properties of the membrane, the width of the membrane, the quality of the membrane, it's rather universal. It's around 1 microfarad per centimeter square. So this is rather universal, but this is really changing. Rm could change in a wide range of parameters, and this is because this Rm is determined by how many open channels, ion channels, you have in this membrane at rest. So if you have a lot of ion channels open at rest, you will have a leaky membrane, which means a low resistivity, high conductance, low resistivity. So maybe 5 ohm centimeter square is a membrane that has a lot of open channels already at rest. And the high resistivity membrane, sealed membrane, almost very sealed membrane, semi-sealed membrane, has a high number here. And the membrane here in the cortex of a regular cortical neuron will be about 20,000. So it's leaky, but not too leaky. But the multiplication of these two, which is the most fundamental passive membrane property of neuron, is this tau, this time constant of the membrane, Rm multiplied by Cm, which in my case, when I mentioned 20,000 for the cortical cell, it will be 20 millisecond time constant. The property of the membrane, how fast the membrane charge and discharge, depends very critically on this tau m. Okay? So 20 milliseconds. Sometimes very fast membranes, because it's very leaky. So the time constant is really influenced by the quality. Of course, it's a multiplication of these two parameters, but this is rather fixed parameters, so this is really what fixing, what causing a membrane to be more or less leaky, or faster or slower time constant, which is the same thing. That's a very fundamental property. It means how fast you can really take your input and build up a voltage, and how much you can get rid of this voltage after the ending of the input. It's a very fundamental property of the membrane of neurons, which you really should understand. I'm sure you do, because you learned a little bit about membrane circuits. Okay, so these two, of course, there is another important parameter, which is the axial resistivity. Because you have a cylinder, you have a cable, there is a membrane wrapping it, but there is this axial resistivity, the cytoskeleton, inside the inside the volume, and this is sometimes called Ri or Ra, the axial resistivity per unit volume. And so these are these numbers, 70 to 250 ohm centimeter, not centimeter square, ohm centimeter. So these are really the three basic, if you ask me what do I need to do in order to characterize a neuron, the passive properties of neuron, I should say you should know Rm, you should know Ri, or Ra, the axial resistivity, and you should know Cm. The most fundamental thing you can say about the cylinder, passive cylinder, you should add also as another characterization the input resistance of the system, R input or Rn, which is this quality which depends also on morphology. Yes, the morphology, because these are all specific numbers. These are per unit area or per unit volume. But eventually you have a real structure with a given dimensions. It could be small cell, it could be big cell. With the same membrane properties and the same axial resistivity, you will get different input resistance. So if you have a big cell, you will have low input resistance. So input resistance is another characterization of the cell. Because when I am a synapse and I impinge on the cell and I inject this amount of current, how much voltage will be developed depends on the input resistance, actually on the input impedance, because it's not a steady state input. But this input resistance is very fundamental property of cells, and in the cortex you should remember maybe 100 mega ohm. 100 mega ohm. Mega is 10 to the power of 6. So it's a very, very, very large input resistance, which means that you need very small current from the synapse to develop large voltage, relatively large voltage. Very small current.
Rm dependent only in the density and like number of channels, or it's dependent also in specifications of the channels?
So of course, so this you should think about an ion channel embedded in the membrane as a small resistivity, and another one in parallel, another one in parallel, many, 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 many parallel resistance. So the more you have, more shunted, more current flow can go through the membrane. And of course, the property of the channel, the conductance of a given channel, of course, determines this R of the channel. So some channels have a larger conductivity, some have a smaller per channel. But typically, again, if you want to go to channel level, a channel will have 10 nanosiemens conductance. Nano is 10 to the power of minus 9, yes? 10 nanosiemens, Siemens is 1 over ohm, yes? 10 nanosiemens conductance per channel. No, there are some other channels, bigger or smaller. So it's not only the number of channels, but of course the total conductance of channel multiplied by the number of channels per unit area will give you this number. Okay? I'm not repeating something that you already know because this is really, really, really fundamental. You know, very good. Excitability is something that you should add on top of it. So channels, of course, are voltage dependent, and there are many, 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 many soup of channels. And the dendrites are renowned for having most of the nonlinear channels in the dendrites are in neurons are in dendrites. So it's interesting to think about the fact that the cell body generates proteins all the time, the machinery generate proteins. These proteins are being transmitted, so to speak, to here, to there. And most of these proteins, which become channels, are being embedded to the dendrites. There are many, many tens of types of calcium channels, tens of types of potassium channels, many types of sodium channels. And we know today more genetically, you know, the composition of a channel. And it's interesting to know that you put so much effort, not only in terms of synapses on the dendrite, but also in terms of nonlinearity in dendrites. You put a lot of effort to send channels to particular regions in the dendrites. Must mean that the dendrites are doing some interesting nonlinearities. Okay, so on top of this, you have these nonlinearities, which some of them generate full-blown action potential in the dendrites, the calcium spike. Some of them enable this backpropagation, the sodium channels in the dendrites, the voltage-dependent sodium channels in the dendrites. But some of them are doing little modulation. Most of them are not dramatic action potential, but they are nonlinear in terms of summation, in terms of activity. And so when I say there are many channels, I mean that there are many dependencies on time and voltage of actually 4 types of channels. The calcium-dependent, voltage-dependent calcium, sodium-dependent calcium, potassium-dependent calcium, maybe a little bit chloride. So all these channels carry all these only 4 ions. But the dependence on voltage, the time constant of the channel, how long it opens, how long it closes, how fast, all this is this inside here. When I say excitability, I mean all this. And this is a big enigma, exactly, to characterize the type of ion channels in dendrites, because this is the resolution of membrane pieces, so to speak, patches, and this is not an easy characterization, but we understand that there are very nonlinearity, a lot of nonlinearity in the genetic tree, not only passive properties. But cable equation deals only with this part. A nonlinear cable is very difficult, of course, to mathematically characterize or analyze, and so we are using numerical method. We'll talk about this. And of course, there is this synaptology. I spoke about morphology, about physiology. I should speak about synaptology. And this is exactly the issue of how many synapses and what is the geometry of synapses. For example, the geometry of synapses is such that when you have spines, most of the time you have excitatory synapse on the spine. Rarely you find spines with 2 synapses, then you find them inhibitory and excitatory on the same spine. So the geometry of connectivity is not random. As I said, when you have spines, it's excitatory mostly. Inhibition goes to the cell, to the basis of the dendrite, not to the spine. Mostly to the soma, near the soma, axon initial segment. So there is some geometry of these synapses. They are not always everywhere. And in recent years, and that's very important for people trying to understand single neuron computation, we find in recent years, because we now have all these fantastic genetic tools. So this is, let's say, layer 5 pyramidal cell. And you ask yourself, what is the geometry of synapses impinging on this, connecting into this dendrite? You can see, for example, that basket cell, so this is one type of pyramidal cell, of inhibitory cell. So this is the basket cell, is making mostly synapses near the cell body. So this is the territory of the basket cell type, not only one basket cell. The basket cells population targeting inhibition near the cell body. There is another cell, Martinotti cell. So this is another inhibitory cell. So this cell likes to target mostly here, let's say, distally, inhibitory. So there is a sharing of labor, interesting sharing of labor, which we do not understand. That's another enigma. There is kind of a sharing of labor between all these subtypes of inhibitory cells. There are subtypes characterized genetically or chemically. Some of them are parvalbumin type cells, inhibitory, that has parvalbumin within, so you can stain them for parvalbumin. Some of them are from other chemicals, so you can characterize them not only by structure, but also by chemistry. So why is there such a kind, why is there sharing of labor? Why don't I just inhibit everything? Or not everything. Why don't I just inhibit the initial segment of the axon? Because after all, if I'm inhibition, if my claim to fame is that I can kill the axon or not kill the axon, I'm in the spine. I can inhibit or not inhibit. Why do I care to put inhibition so distally from the output side? What is this game between inhibition and excitation? We don't completely understand this kind of game. But we understand that there are two types of synapses in general, excitatory and inhibitory. A given cell receives mostly excitatory in the cortex. So 80% of the synapses, 90% of the synapses are excitatory. Only 10% of the synapses are inhibitory in terms of numbers, not strength, number. But they are not, the inhibition and probably the excitation are not randomly distributed. Different sources, different type of cells target different dendritic regions. There is a territorial issue here which we don't understand functionally. So when I speak about synaptology, I mean also this. Not only numbers, not only type, not only strength of inhibition, not only time course of inhibition, but also territory of inhibition. Because apart, they are very, very distinct. For example, there is one most distinct inhibition, most particular inhibition, and this is called the chandelier inhibition. Chandelier. It has a very nice structure, this cell, like a chandelier, the dendrite of this cell. And it goes only and particularly and specifically to the initial segment of the axon. So this particular chandelier inhibition goes specifically there. It doesn't make synapse in the dendrite, sometimes one or two in the soma, but mostly it really covers, tuck, 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 the initial segment. It likes to control the initial segment. So you can see the specificity of this territorial issue. This is the most specific, because others cover a more bigger region. And so there is a recent paper, and I spoke about this last year from a student of mine who is now in postdoc, Albert, in Huron. We are very proud of this paper because this paper tried for the first time to try to give a sense to this kind of territorial distribution, biophysical sense. But I'm not going to talk about this, just for you to know. Okay, and just another thing you should know, maybe you know, you should tell me, there are these two very broad type of inhibitory inputs. GABA, by the way, is the most prominent in the cortex transmitter used for inhibition. So presynaptically in the axon, there are these gamma aminobutyric acids sitting within these vesicles. But postsynaptically, there are two type of receptors, the GABA A and the GABA B. You know about this GABA A and GABA B. The important thing about the time course of them, they are very different time course. This is a fast inhibition, and this is a slow inhibition. This is based on chloride. This is based on potassium channels, but as you know, I'm not going to talk a little bit more about that. Okay, let me put you to another controversy. Controversies are very important for science, as you know. You don't want to agree on anything, and you should not. Neither with me, of course. You absolutely should not accept what I'm saying as any truth. It's a tentative truth. I hope it's more than a tentative, but there are things that will be developed. But anyway, so there are controversies because people do not believe there are very strong views, and these two really fought terribly about the following. So Eccles is a Nobel laureate, John Eccles. He was working mostly on spinal cord neurons, and he analyzed all his data along the years, in the middle of the 20th century, from the 40s onward. Eventually got Nobel Prize for fantastic work on spinal cord and motor control, movement. And he characterized, the reflex, and, the reflex when you go to a physician checking your reflex and all that. But he also measured directly from spinal cord neurons, and he assumed that these neurons are isopotential in terms of the fact, or he assumed that dendrites, I will talk a little bit about that in a second, dendrites are not important in terms of analyzing the signal that you see at the cell body. He said for a very good first approximation, I could assume that dendrites are isopotential, that neurons are isopotential. RC circuit. Roll, who was his postdoc, said it cannot be. I can see all this structure. I can see all this dendritic structure. Cajal, beautiful neurons. I record from the cell body. I get a signal. I get an EPSP, or I get a signal from injecting a square pulse into the soma, and after this square pulse, the voltage attenuates, decays. It cannot be one exponent. One exponent means isopotential. So they really fought hard. Again, I don't know why scientists are so brutal sometimes, really brutal. So Eccles said this is a good approximation. And it could be good approximation to certain things. In all books you don't want to have all this big picture. It takes a lot of the page, yes? So you reduce the complexity of describing a neuron like this. And this is exactly what Eccles had in mind in terms of model. He said, I could assume that all the synapses that are relevant, the nearby synapses, sit in the cell body. The distal synapses, I know that they are there. He knew that they are dendrites, of course. But they are so far they don't really influence the cell body. Let's leave them. But the main thing is that let's analyze the signal in the cell body as if it is coming from an isopotential membrane, from an RC circuit. So this was the controversy. It was a very big controversy. I don't want to tell you all the story because Roll was my postdoc supervisor, really suffered from him all his life in some sense, because he did not allow roll cable theory, which is the fundamental of what we do today in terms of understanding dendrites. He did not allow it to be published. So suppose you are my student and I don't like your theory, I don't allow it to be published. Very totalitarian. And so he didn't allow it to be published. What does it mean didn't allow? Whenever it was sent, they asked Eccles, what do you think? He said it's bad. So it was not published until Roll found a little journal that did not know about Eccles, or the Eccles did not know about the journal. He succeeded to publish it there. This is a little bit of a gossip from science. So Roll said, okay, so suppose I inject a current to the cell body. Let's say square pulse. From my simple, on paper calculation, most of the current that I inject will escape to the dendrites. It will not go through the membrane of the cell body. Just because conductivity, if you compute very simply, with a cell body, with this membrane properties, with this Rm, with this Cm, yes? If I had a cell body, but this is a dendrite with this diameter, this, yes? I just compute what would be the axial resistivity, physical, not the specific one, the real resistivity in ohm, here. I see that most of the current will escape there. It will escape there. So how can I assume that it's isopotential? It's not, something is wrong. But Eccles did not like it. Because, for example, one basic thing you want to know about neurons is the time constant of the membrane. That's what we discussed, the time constant of the membrane. So how do you compute the time constant of the membrane experimentally? You take an electrode, you inject a little square pulse, as we discussed, or a little delta function, small current like this. Okay, so you go into your cell body. There are dendrites, but you have your electrode in the cell body here. You inject a square pulse here, or a very brief pulse, doesn't matter. And you look at the voltage attenuation following the ending of your square pulse. So let's say you finished here. There will be an attenuation. If you assume it's isopotential, because it's isopotential, there is mostly membrane of the cell body, but not elsewhere, then if you would plot it on a semi-logarithmic scale, this would be ln V, and this will be T, and this is isopotential, you will get a straight line. because this is E over minus t divided by tau, yes? This is proportional to that. So if I take the ln of that, I should get a straight line. And the slope of this line is or 1 over the slope of this line, minus 1 over this line is my time constant. So this is how people today analyze the time constant of their membrane. But Roll showed in his cable theory, which you know already, at least some of it, you know that if you have a distributed system, a distributed electrical system rather than isopotential electrical system, which means that there are differences in voltage between where you inject and elsewhere, which means that there is axial current, as you saw here, axial current from the source to the sink. This axial current and the fact that you have cables there means that there are extra time constants beyond the membrane time constant. This means that the attenuation here is not governed by your membrane time constant. It's governed by the time, by the actually infinite number of time constants which come out from the cable equation. And so it should decay like this, something like that. So if you now plot carefully, not if you had a model in your brain and you put your model and you ignore the fact that actually the attenuation is like this. You said, this is about 1 exponent. But it's not 1 exponent. So if you say that this is 1 exponent and I take the slope here, let's say, I get a much slower time constant, much faster time constant, actually the membrane time constant. Because if you think about this like Roll did, and you wait enough in terms of time, even your non-exponential time constant, or non-exponential voltage decay, if you wait enough, the system will become isopotential. And this is an intuition I want to give you. Again, you may have heard about this, but why a system becomes isopotential if you wait enough? Because when you inject a current here, and you build a voltage, and you stop injecting, I stopped injecting, so I generated locally large voltage here at the soma. There is no voltage yet, but I stopped injecting. Now there is a current flow from the high voltage region to the low voltage region. So what happens is like this. My soma goes down. I lose current, both axially and also through the membrane. I lose current, but voltage there grows. Yes, it does like this, until it becomes isopotential. And then there is no axial current anymore because it's isopotential, and then I always only go through the soma. I lose the current to the membrane resistivity. Okay, so I do like this, and then I go through the membrane, and then it's isopotential, because isopotential means that only the membrane now controls, because the membrane the membrane itself has path for current, yes? So eventually, when everything is isopotential, I can consider the system as isopotential. It is functionally isopotential, and when it's isopotential, the slowest decaying time constant, and that comes from Roll's theory, the slowest decaying time constant is the membrane time constant. And this I have to prove, that other time constants are faster. What Roll called them the equalizing time constant. How fast I equalize the voltage from here to here. These are the equalizing time constants. There are infinite number of them. There is one slowest one, the membrane time constant, and it's here. So I should peel the time constant, that's how we called it. I should peel the time constant from the tail of my decay, from the tail. And if I do that, I get the correct time constant of the membrane, Is that clear? So that's what Roll did. He said, you are wrong, because most of the current is flowing. There is a distributed system, just I can see. I assume that it's not isopotential. That's why it has infinite number of time constant. Wait, peel the tail. Look at the attenuation at the tail. And that's what we do today. After this theory, after a neuroscientist who wants to you ask him, what is the time constant of your membrane? He does this, ln V versus T, but he looks at the slope far from the beginning. because here the slope is faster time constant. And if you wait enough, you get this. So this is minus 1 over tau. From here, you get the tau 0, or what is called the tau m. And this is the correct time constant, which is larger than what Eccles measured. And this was one of the big controversies, because Roll said you estimated the membrane. I told you membrane time constant is a very important parameter to characterize cells. And so what is the correct time constant? So Eccles said in the motor neurons it's 5 milliseconds or something like this, and Roll said, no, it's 10 milliseconds. It's 10 milliseconds. It's slower than what you thought. And this means that Nobel Prize made a mistake. He published wrong numbers. This is something you don't like. Just because you had a model in your brain that the cell is isopotential, you adjust your analysis to your model, you get a wrong number, you stick to it, and somebody comes with a wrong number, with another number. It means that you published 5 papers with wrong numbers, which is not good for a non-laureate. Anyway, so that's just an example of why you need correct model to analyze your data. I gave you an example. So just to finish with Roll, and then I'll let you go, and we'll meet on, when is it? Wednesday, no? Or Tuesday? Wednesday, no? Wednesday. Okay, so this was Eccles. When I said this was Eccles, I mean this point neuron. Actually, it's much before Eccles, of course, McCulloch and Pitts. I hope you heard about McCulloch and Pitts. And if not, we'll talk a little bit about, because they were introducing computation to neurons, but they assumed this point neuron, which means you collapse, you neglect all the dendrites, and then it means also that you analyze the signals, as I showed you, erroneously. You can say that it's not important. Who cares about 5 or 10, and that's fine. But if you want to get the correct biophysical numbers, you have to have the correct model. So that's one aspect. And then the question, and that's an ongoing, of course, question, is do we need, for what purpose, to take the real morphology, so to speak? So if I want to build a model of neuron in my future robot, do I need to take all the fine details with all the spines and all the synapses and, copy the neuron in all its fine details? Or can I do some systematic reductions to get something that still holds some important property, and depends what you are interested in, of course. But you don't want to lose something fundamental. And of course, you don't know what is fundamental in advance. But on the other hand, as a physicist, you don't want to take all the details in advance, because then you don't get the principle. So that's the tension. I would say the tension is in neuroscience, is really here. At what level of models are correct for a particular question you want to understand? There is no general answer, of course, because it depends on the question, yes? If you ask about a single spine, the level is a single spine, or below. But if you ask on a single cell or a network of cells, dynamical networks, what levels do I need to take? Not only in terms of morphology, but also in terms of electricity. Is integrate and fire model is a good representative of real spikes or not? In what sense? So Roll suggested actually to do the following, and this was his paradigm. He said, take the simplest, go to find for a particular question. Take the simplest and see what you lose or what you earn. What you lose or what you earn if you go and make it more complex, until you don't get more benefits, so to speak. So you can go both ways, either this way, and eventually you get to the full model, which is very cumbersome mathematically. We can do it today with neuron models and others, neuron simulators, which I'll talk about that. And eventually you get a model, which is always a model. You don't take the real cell in your model. You take a model of the cell, so it's a model, and you get the kiss. But in this case, this is the detailed kiss of Rodin. So it's a model. It's not a real kiss of a couple, but it's a full detail. To a certain degree, it's a model of a kiss, beautiful kiss. So you go to Paris, you go to Rodin Museum, you see this kiss. And I think the theoreticians, the abstract theoreticians like Chaim and Tali will say, I don't want this level of kiss because I want to understand kissiness, but not this kiss and not detailed kiss. I'm doing the opposite. I'm going to the opposite, and I want to capture the kiss like Brancusi. So this is the most minimal kiss. Yes, this is the point neuron kiss. Because if you reduce it a little bit more, then you lose the kiss. No kiss anymore. Okay, that's just a metaphor that I like to use because I like not only kisses, but it is a sculpture there. I like these two. By the way, both are in Paris now, so I mean the museums. The Atelier of Brancusi in front of Pompidou, and this is in Rodin. So, but I think that's the tension, yes? What level for a particular thing? So if you just want the simplest kiss and the model of a kiss, and you want to understand whatever it means to have a kiss, that maybe this is good. Certainly beautiful. But if you want maybe to understand more about details and about interactions, maybe you need this level of description or this level of description. So we'll talk a little bit about this level of models and what role was suggesting for a particular question. I showed you one example. But if you want to understand the signal in the cell body, this is not an appropriate model. But we don't need all this model. We all showed how you can simplify this to something like this and even simpler, but still have a structure. And you get most of your behavior in terms of the cell body. Okay, so let's wait for next Wednesday, for the coming next Wednesday.